Okay, Assalamualaikum warahmatullahi wabarakatuh. I am Nur Huda binti Harudin from the second year Marine Science, and today I will explain about the diversity of the marine phototroph. Okay, before we start, we need to know what is the meaning of phototroph. Okay, basically phototroph is an organism that synthesizes ATP by using light as their energy sources. Then, this organism synthesizes ATP by ATP synthase and using a transmembrane electrochemical potential by pumping up a proton from a membrane one side to another side based on their light energy. Okay, now we move to another chapter, which is timeline. Okay, in 1970, the primary producer that was found in that year is diatom and dinoflagellate by using a plankton net. But in late 1970, they found another bacteria, which is cyanobacteria, by using a fluorescence microscopy. In late 1980, they found another bacteria, which is picocyanobacteria, by using HPLC. But in 1996, they found chlorophyll D. In 2000, they found the AAPB and rhodopsin. In 2010, they found chlorophyll F, and 2011, they found UCY and A. Okay, now we are in traditional understanding of the primary producer. Okay, as I said just now, in 1970, the primary producer that was found in that years is diatom and dinoflagellates. This is because these two plankton is a major collected by a plankton net. Okay, these two planktons can be found in eutrophic and oligotrophic region. Eutrophic regions mean the region which is high nutrient, and oligotrophics mean low nutrient region. Okay. Plankton nets, it has a three size start in 1970, which is 100 micrometer, 70 micrometer, and 60 micrometer. Okay, basically the small mesh size of the plankton net that we use is the small size of plankton that we can get. Okay, now we move to the recognition of the Picocyanobacteria dominans. Okay, in late 1970, the primary producer that was found in this year is Cyanobacteria by using the fluorescence microscopic developments and the accumulation from the knowledge electron microscopic observation. These tiny cells, which length 0.5 until 2.0 micrometer in length and 0.5 until 1.0 micrometer in diameter. While in 1980, another bacteria was found, which is Picocyanobacteria. Picocyanobacteria was developed from the HPLC developments. Okay, HPLC means the high performance liquid chromatography for pigment analysis and use flow cytometry for the cell detection. Each cell contains a dividend chlorophyll slightly different from a chlorophyll A and didn't contain picobilly pigment. Okay, this cell around 0.5 until 0.6 in diameter and 0.5 until 0.8 in length. They form oxygenic photosynthesis by using divinyl chlorophyll A or B by absorb the blue light dominating in our ocean. Okay, in several regions, the amounts of the divinyl A is much higher, much higher than the chlorophyll A in some regions. Okay, now we move to the discovery of ubiquitous phototroph. Okay, in 2000, another two bacteria was found, which is N-oxygenic photosynthetic bacteria, which is that we call AAPB and Phototroph. Okay, for AAPB, it contains a bacteria chlorophyll that's recognized as a pigment photosynthetic pigment in anaerobic and oxygenic photosynthetic pigment, which is ANAPB. Okay, ANAPB is anaerobic photosynthesis. ANAPB reach 30% of primary production in some slate, while AAPB you grow photoheterotrophically in anaerobic environments. AAPB use oxygen as their growth and bacterial chlorophyll for synthesis. AAPB distributes widely in euphotic zone of marine environments. AAPB grow faster in a dark, and AAPB in seawater was faster than other heterotroph bacteria. Okay, for the proteorhodopsin, rhodopsin is like proteins that detect in a marine proteobacteria. Rhodopsin ni are like photoreceptors that known as a light sensor. It contribute to synthesize ATP by using a bacteria rhodopsin. This I show how the reaction occur in a membrane cell, and many type of the rhodopsin can be found by using a metagenomic analysis. Okay, now we move to oxygenate photosynthetic using the far red light. Okay, basically we just know about the chlorophyll A. Okay, basically chlorophyll A ni for the photosynthetic pigment that we familiar is the absorption range dia ialah 400 nanometer until 700 nanometer or 380 nanometer until 710 nanometer. Just is defined as a photosynthetically active radiation. But okay, in 1996, another bacteria was be found, which is chlorophyll D. In chlorophyll D, it can absorb far red light, can absorb 13 nanometer wavelength more than chlorophyll A. Okay, the energy transfer efficiency between chlorophyll D and chlorophyll A is same. The amount of chlorophyll D in seaweed is more than chlorophyll A in a range 1 until 13%. Okay, the latest chlorophyll that we found is chlorophyll F in 2010. Okay, this chlorophyll that was found in Australia in cyanobacterium. Okay, in chlorophyll F can absorb more the wavelength than chlorophyll A, chlorophyll D, which is it can absorb 720 nanometer wavelengths and absorb far red light until 760 nanometer in vivo. Okay, now we move to the discovery of eukaryotic phytoplankton. Okay, in 2000, species of Chlorophyta heterophyta is known as a eukaryotic phytoplankton. In 2010, metagenomic technique and the flow cytometry were published. Okay, 19HF is predominant detects in a picoplankton fraction less than 3 micrometer at the high latitude area in a 40 degree until 60 degree or of both north and south. Okay, while in 2007, the phytoplankton which didn't contain to any phyla of the algae is discovered by the metagenomic analysis. Metagenomic analysis is like using a DNA analysis. Okay, this is a photo of the process of metagenomic technique. Okay, while in 2011, the new candidate phylum was detected by the metagenomic analysis targeting plastic DNA, which is Rapamine. Okay, for strange phototrophic microorganism, the cyanobacteria like UCYNA was detected in a metagenomic technique of nitrogen fixation in a marine water. Okay, the wide distribution of UCYNA indicates that the particle contribute to the nitrogen cycle in a pelagic water. Okay, UCYNA ni actually dia undergoes photosynthesis 1 and dia tak undergo photosynthesis 2 pun. Photosynthesis 1 ni dia mesti ada chlorophyll yang untuk dia bergerak semua tu. Next one, we move to the diversity of the light energy transformation system and reconsideration of a photosynthesis. Okay, in picture 1, we can see proton was pumped into a tracheate lumen from the stroma, and we can see the ATP synthase happen in a stroma, not in a tracheate. While in B, we can see the proton is pumped out from the lumen, from the tracheate, and the ATP synthase happen in a lumen. This is because of B is a non-oxygenic photosynthetic, while A is oxygenic photosynthetic. Okay, the example of bacteria in A is cyanobacteria, while in B is Proterohydropsin.